Hello, this is Ed Chapman, and this video cast is going to focus on ion channel receptors, another setup for cell signaling, especially the reception part of cell signaling. Now, before you proceed, make sure you understand the video cast that came before this one. Can you answer these 4 questions? If you can, then I think you're ready to go on to the next situation. All right, ion channel receptors are probably the simplest of the 3 different forms of reception we're going to focus on in this chapter. They're also known as ligand-gated ion channels. And the reason why I think these are simpler is because they're all about regulating facilitated diffusion, which we've already talked about. Now, just to get ourselves oriented here we have a phospholipid bilayer, or a plasma membrane. Here we have the cytoplasm of a cell, and here's the outside of a cell. Now, outside of this cell, there is a high concentration of ions, and the only way these ions can get through the plasma membrane is by going through an embedded protein. But this embedded protein is closed, or it's gated. It needs to be opened. The gate needs to be opened, and this gate can be opened by a signal. Okay, now we've seen this before. This signal molecule, called a ligand, is going to interact with the active site with an active site on this embedded protein and cause it to open. So we go to the next slide. You can see that the signal has been received. We have a conformation shape change. The gate opens, and now the ions can flow down their concentration gradient. And because this is diffusion and it's a short distance, this can happen very fast. Okay, and it's a way for the cell to regulate facilitated diffusion. Now, if we go to the next slide, we notice now that the signal molecule has disengaged, the gate has closed, and whatever ions have gotten into the cytoplasm of the cell, they're there, they can't get back out, and most importantly, the gate's closed, so no more can get in. So in order for this system to reset, these ions down here that have already gotten in are going to have to be pumped out. Okay, remember the sodium-potassium pump? That's one way we could reestablish the gradient that started this whole signal pathway. Okay, do you see how this stuff fits together? That's why I think it's really interesting. All right, now why is this important? Well, gated ion channels and the signal molecules that activate them are key to understanding how our nervous system works. Okay, nerves communicate with tissues and with other nerves with hormones called neurotransmitters, which function over a very short distance. And because they're bonding to receptor proteins on target cells, they're ligands, and these receptor proteins are ion channel receptors. Okay, and once they open, they allow ions to flow into the target cell, and the change in ion concentration can be a way to trigger a nerve impulse. Okay, let's look at a picture of this. Here we have a closed or a target cell that has not been switched on or activated. Here we have a nerve cell that is releasing a neurotransmitter, which is going to function as a signal molecule. Notice that when the signal molecules bond to this receptor protein, which is a gated ion channel, we now notice that it opens up and ions can travel into the target cell, and this can maybe generate another electrical impulse or nerve impulse traveling somewhere else in the tissue or in an organ or even in the whole body. Okay, so what I want you guys to be able to connect here is connecting it to the nervous system. The nerves release neurotransmitters. Okay, the neurotransmitters diffuse as signal molecules attached to a embedded protein that's functioning as a facilitated gate, which can be open. And once they are opened, because they've received the signal, they can allow ions to pass through into the cell, and hence we have a response. All right. We'll stop there. The next video cast is going to explore the second part of cell signaling, which is signal transduction. Thanks for listening.